Hello students, let's start today with another important question of paper 1, chapter 1, reproduction in lower and higher plants. The question is, explain the development of endosperm, or explain the different types of endosperm. Different types of endosperm have been asked in board exams couple of times. So either it could be for 1 mark, what is endosperm, or it might be a long answer on development of endosperm, or explain different types of endosperm. Let's start and understand what is endosperm. Now, if you remember, you have learned about triploid primary endosperm, that is PEN. So, this triploid primary endosperm nucleus repeatedly divides mitotically to form nutritive tissue called endosperm. So, when this pen divides mitotically to form nutritive tissue, it is called as endosperm. And it is a post-fertilization event. The post-fertilization changes within the ovule, embryo, and endosperm. These changes are seen to develop simultaneously. The other cells of embryo sac disorganized sooner or later. The formation of triploid endosperm nucleus triggers cell division, which leads to the formation of endosperm. There are 3 types of endosperms on the basis of mode of development. These are, first one, nuclear type, cellular type, and halobial type. Let's see the first one, nuclear type. It is the most common type found in 1061 angiospermic families. The primary endosperm nucleus repeatedly divides mitotically without wall formation to produce large number of free nuclei. Look at the diagram. Look at the diagram. Primary endosperm nucleus repeatedly divides mitotically to produce large number of free nuclei. See the pen, how free nuclear mitotic division, many nuclei, this gets arranged near the side of the wall, which leaves a vacuole in the center. Okay, and then there is a cell formation around the nuclei. This is called as nuclear type. Okay. So what is the nucleotide? What happens in nucleotide? Primary endosperm nucleus divides mitotically. Large number of free nuclei are produced. Vacuole appears in the center of the cell, and later walls develops between the nuclei. This is termed as nuclear type. Okay. It is the most common type found in 161 angiospermic families. Primary endosperm nucleus repeatedly divides mitotically without wall formation to produce large number of free nuclei. A big central vacuole appears in the center of cell, pushing the nuclei towards the periphery. Later wall develops between the nuclei. Hence, multicellular endosperm is formed. It will be very easy for you to remember the answer if you look at the diagram and will learn to write the answer. So, memorize the diagram and try to learn the answer. Now, let's see the next one. Before we go ahead, let's see the example. In several cases, cell wall formation remains incomplete. For example, in wheat, sunflower, coconut. Coconut has multicellular endosperm in the outer part and free nuclear as well as vacuolated endosperm in the center. So, the coconut water, what it is? It is the endosperm. Okay, that is the endosperm, which are free nuclear. Now, let's see the second type, that is the cellular type. In some plants, division of triploid primary endospermic nucleus is immediately followed by cell wall formation, so that the endosperm is cellular right from the beginning. It is mostly observed in 72 families of dicot. So, primary endosperm nucleus immediately undergoes nuclear division followed by wall formation. So, cellular endosperm right from beginning. Examples: balsam, petunia, adoxia. So, these are few examples of cellular type. And remember one thing, it always starts with pen. Pen is what? Endosperm, primary endosperm nucleus. Third one. Now, this is ideally seen in monocotyledons. Earlier, the first two types that we had seen was found or is been found in dicots, whereas this halobial is found in monocotyledons, order halobials. First division of primary endosperm nucleus is followed by transverse wall which divides the cell unequally. Look at the diagram. Pen, first division divides the cell, okay, look over here, divides unequally, divides the cell unequally. Look at the diagram. Pen, first cell division, divides the cell unequally. Larger micropylar cell and smaller chalazal cell. Then the nuclei in each cell divide by free nuclear division. Then walls develop between nuclei in micropylar chamber. It is intermediate between cellular and nuclear type of endosperm. So, once again, look at the diagram. Wall develops in between nuclei of micropylar chamber, while in chalazal chamber, okay, what happens in chalazal chamber? It remains like this. So it is a combination of cellular type and nuclear type. So it is termed as halobial type or an intermediate between cellular or nuclear type of endosperm. Example, asphodelus. Okay. Look at the diagram and draw this diagram in your notebook. All the three types. Now let's see the next question, that is development of embryo, in the next class. So for the time being, you will learn what is endosperm, definition, and the three types of endosperm. So what are the three types of endosperm? The first one is the nuclear endosperm. Next one is the cellular endosperm, and the third one is the halobial type of endosperm. I'll take you through the slides once again. See, nuclear type, cellular type, and halobial type. Write this in your notebook and complete this one short answer. Thank you, students. Take care of yourself. Stay home and stay safe.